Hello everyone, my name is Dawar Khan and welcome to my talk on Dr. Kit for Scalable Physicalization of Organic Shapes. This is an agenda of the talk. I will start from research history leading to this project and motivation behind this project. Dr. Kit helps in physical visualization of the microscopic biological structures. Visualization of the mesoscale biological structures helps in understanding their functionalities and behavior. It provides a baseline for further predictions of these structures. In the COUS Nano-Visualization Research Group, we are working on such type of research, which includes visualization of these structures at atomistic level, their modeling, rendering, and prediction. We are also working on generating virtual environments of these structures and providing guided tours with narrated documentaries of the virtual environments. Now, the question is why not to have a physical touch to these objects instead of a virtual view? Such type of physical models can play an important role in education and learning, as it provides a clear abstraction of the shape and can increase the user interest. These models can be used in scientific museums, awareness seminars, educational institutes, and scientific conferences. In our context, we are interested in physical visualization of a cute family of these structures having potato-like shape with a closed membrane around it. Instead of a full membrane, we are interested in its decomposition and modeling with reconfigurability in a puzzle-like fashion. The puzzle improves memorability and engagement and provides us a learn with fun opportunity. The modeling is based on isometric decomposition, which means modeling with only k types of triangle. For example, this model has only 2 types of triangle. Such type of models increases usability and thereby reduces the cost. This is an overview of our method. On the left side you see the biological structure, and on the right side you can see the corresponding physically printed models. We are interested in modeling these structures with at least 3 conditions. Modeling with only k types of triangles should support reconfigurability in a puzzle-like fashion and must preserve the input shape. The reconfigurability is achieved by holes and hinges. The intermediate steps are discussed one by one in the upcoming slides. The first step is mesh generation, where we use marching cubes algorithm to generate surface mesh of the biological structure. The raw mesh is then improved via CVT. CVT is an algorithm that improves the mesh regularity by moving each seed to the center of the corresponding coronae cell and thereby improves the coronae tessellation and the triangulation. We can also set the number of vertices here to minimize the complexity of the mesh. Clustering. Clustering is the key contribution of our work. Here we use k-means clustering to classify the triangles into k classes. For such type of classification, we need to find similarity among the triangles. We use a simple distance function for this similarity calculation. For this purpose, we map all the triangle into a 3D space. Each triangle is mapped into a single point in a 3D space such that the shortest edge is mapped as a distance from the x-axis, the middle edge is a distance from y-axis, and the longest edge is a distance from the z-axis. In this way, all the triangles are mapped into 3D space such that the distance between the corresponding point indicates the degree of dissimilarity of the two triangles, and then we apply k-means clustering to classify these triangles into k classes. This leads us to this equation. After clustering, for each cluster or each class, we find the center triangle Ti steric, and all the triangle in the same cluster needs to be identical to this triangle. So for each triangle Tj of the same class, we find the distance d, which represent the degree of dissimilarity from the center triangle to any triangle Tj. Then we apply remeshing operators to minimize this distance, these individual distances, which minimize the overall cumulative distance. Remeshing. As stated in the previous slide, remeshing is used to increase similarity among the triangle or to minimize these distances. We use three types of operators. H-collapse and H-flip are executed only once, while the vertex translation is iterated with clustering until we reach the desired number of k types of identical triangles. For vertex translation, we need new position where the vertex needs to be translated. Based on similarity with the center triangle, we calculate the required edge length for all edges. It puts pressure on adjacent vertices either inward or outward direction depending on whether the required edge length is longer or shorter than the current edge length. Then for each vertex, we find the average pressure from all adjacent edges, and the vertex is translated to this new position if it doesn't destroy the input geometry. After getting the k-set triangulation, the next step is thickening. You can see the cut view surface mesh has no thickness. We thicken the mesh in the direction of the vertex normal. These models shows thickness in perpendicular extrusion on the left side and our method of curvature-aware thickening on the right. After this, we created holes in these triangles and design unique hinges for their assembly and reconfigurability. As an additional step, we also have smoothing with subdivision to get smoother results. The triangle subdivisions provide us curved faces instead of the planar triangle. For the curved faces, we use an additional fourth axis to calculate the degree of dissimilarity between the two patches. This fourth axis indicates the curveness of each patch. We empirically evaluated our method on different models and printed some of these models. These are clustering results. On the left column, you see the input mesh. The center column shows the CVT initialized mesh, and the right column is the clustering results, and this column shows the number of classes of triangles in each model. Again, input mesh CVT initialized and after clustering with k is equal to 8. Some more models with input CVT and after clustering with k is equal to 9 and 11. This plot is showing the minimization of the cumulative distance with the number of iterations until we reach a given threshold. The threshold is the highest value of the acceptable clustering error, that is the acceptable degree of dissimilarity. Here are some printed results. Here the left column shows the virtual mesh, These are the clustering results output, and the right column shows the corresponding printed models. Two more models with the left the virtual mesh and the right the physical printed models. Here is a small comparison with an existing method. You can see our methods has better visual than the previous methods. For more results and especially the quantitative results, please refer to the paper. In conclusion, we used a simple distance metric for K-set isometric decomposition to achieve a cost-effective physicalizations of the biological structures. It supports assemblies and reconfigurability. Thank you for your attention.